Hello students, today's topic is on special proteins and we will be discussing on 3 special proteins, that is enzymes, amyloids and prions. Some proteins have a different or special characteristic feature or function. They are considered special proteins. Some of them are enzymes, prions and amyloids. These proteins have different functions as compared to other proteins of the same class to which they belong. The first one is enzymes. They are a special type of monoclonal antibodies with catalytic activity. The possibility of catalyzing a reaction by means of an antibody which binds to the transition state was first suggested by William P. Jenck in 1969, and later in 1994 Peter G. Schultz and Richard A. Lerner received the prestigious Wolf Prize in Chemistry for developing the catalytic antibodies for many reactions. Coming to the occurrence of enzymes. Enzymes are found naturally in the human body or they can be artificially made. Usually they are raised in laboratory animals which are immunized against a synthetic heptane. But some enzymes are found in patients where the hydrolytic antibodies act against vasoactive intestinal peptide, DNA and thyroglobulin. They are also found in patients with autoimmune diseases such as systemic lupus erythematosus, where they bind to and hydrolyze DNA. Catalytic antibodies which can hydrolyze factor VIII have been discovered in hemophiliac patients. Thus, this particular enzyme prevents the coagulation function leading to the disease. Now comparing enzymes with a normal enzyme. The binding of an antibody to its antigen is similar in many ways to the binding of an enzyme to its substrate. In both cases, the binding involves weak non-covalent interactions and exhibit high specificity and often high affinity. What distinguishes an antibody-antigen interaction from an enzyme-substrate interaction is that the antibody does not alter the antigen, whereas the enzyme catalyzes a chemical change in its substrate. However, like enzymes, antibodies of appropriate specificity can stabilize the transition state of a bound substrate. Thus, they reduce the activation energy for the chemical modification of the substrate. Coming to the mechanism of action of enzymes. We know that the enzymes function by lowering the activation energy of the transition state of a chemical reaction. If an antibody is developed to bind to a molecule that is analogous to the transition state of a given chemical reaction, the developed antibody will bind to and stabilize the transition state just like a natural enzyme, lowering the activation energy of the reaction and thus catalyzing the reaction. By raising an antibody to bind to a stable transition state analog, a new and a unique type of enzyme is produced. For example, a heptane carrier complex was synthesized in which the heptane is a structural analog of the transition state of an ester, which is the substrate, undergoing hydrolysis. Spleen cells from the mice are immunized with this transition state analog and then were fused with the myeloma cells to generate monoclonal anti-heptane monoclonal antibodies. When these monoclonal antibodies were incubated with an ester substrate, some of them accelerated the hydrolysis of the transition state of ester by about 1000-fold. That is, they acted like the enzyme that normally catalyzes the substrate's hydrolysis. The catalytic activity of these antibodies was highly specific, that is, they hydrolyzed only esters whose transition state structure closely resembled the transition state analog used as a heptane in the immunizing conjugate. Coming to the applications of enzyme. First one, enzymes are used in potential HIV treatment. Enzymes that degrades the super antigenic region of GP120 CD4 binding site have been engineered. This is one part of HIV virus outer coating that does not change or mutate. This is the attachment point to T lymphocytes, which is the key cell in cell-mediated immunity. Once infected by HIV, patients produce antibodies to more changeable parts of the viral coat. The antibodies are ineffective because the virus has ability to change its coat rapidly. Because this protein GP120 is necessary for HIV to attach, it does not change across different strains and can be a good target for the treatment. The enzyme binds to the GP120 site of HIV virus and catalytically destroys it, rendering the virus inert, and then attacks other HIV viruses. Number 2, the efficiency of enzyme technology could permit treatments with smaller doses of medicines at lower cost than are possible today. Number 3, enzymes have proven to be very important tool in biotechnology and medical pathology in combating the dreaded human diseases like AIDS, cancer, etc. Number 4, enzymes catalyzing rapid cleavage of hazardous compounds including toxins and drugs such as cocaine. And last, studying them has yielded important insights into reaction mechanisms, enzyme structures and function catalysis and the immune system itself. Now coming to prions. Prions are infectious agents composed entirely of protein material that can fold in multiple structurally distinct ways. The epidemiology of prion diseases is comparable to the spread of viral infection. Stanley B. Prusiner won the Nobel Prize in Physiology or Medicine in 1997 for his research in prions. Now, diseases caused by prions. Human prion diseases include Creutzfeldt-Jakob disease, Gertzmann-Straussler-Scheinker syndrome, fatal familial insomnia, Kuru, transmissible spongiform encephalopathies, bovine spongiform encephalopathy. The most well-known infection caused by prions is bovine spongiform encephalopathy, or also known as mad cow disease, which occasionally spreads to humans who eat infected parts of the cow. The illness is progressive and always fatal. These diseases are also referred to as spongiform encephalopathies because there is progressive deterioration of brain and the diseased brain frequently becomes riddled with holes. Now coming to the structure or isoforms of prions. Prions are made of protein known as PRP, which is found throughout the body, even in healthy people and animals. However, PRP found in infectious material has a different structure and is resistant to proteases that normally break down proteins. The normal form of protein is called PRPC. Here C refers to the cellular PRP. Isoforms of PRPC are PRPSC and PRPRES. The infectious form is called PRPSC. Here SC refers to scrapie, the prototypic prion diseases occurring in sheep. PRPRES may not necessarily be infectious, but it is just protease resistant PRPSC like protein. PRPC is a normal protein found in the membrane of cells, mainly alpha helical structure. PRPC is readily digested by proteinase K and can be liberated from the cell surface in vitro by the enzyme phosphoinositide phospholipase C. PRP has been reported to play important roles in cell-cell adhesion and intracellular signaling in vivo and may therefore be involved in cell-cell communication in the brain. The infectious isoform PRPSC is able to convert the normal PRPC proteins into the infectious isoform by changing their conformation or shape. This in turn alters the way the protein interconnects. Coming to prion propagation. Prions may propagate by transmitting their misfolded protein state. When a prion enters a healthy organism, it induces existing, properly folded protein to convert into the misfolded protein form. In this way, the prion acts as a template to guide the misfolding of more proteins into the prion form. Any mutation in the gene encoding for PRPC, that is the PRPN gene, accelerates the rate of spontaneous conformational change. All known prions induce the formation of an amyloid fold in which the protein polymerizes into an aggregate consisting of tightly packed beta sheets. Amyloid aggregates are fibrils growing at their ends and replicate when breakage causes 2 growing ends to become 4 growing ends. This structural stability means that prions are resistant to denaturation by chemical or physical agents, making disposal of these particles difficult. PRPSC has a higher proportion of beta sheet structure in place of the normal alpha helix structure. Aggregations of these abnormal and misfolded prion protein isoforms tends to form highly structured amyloid fibers which accumulates to form plaques. When one of the amyloid fibrils is broken into smaller pieces, each one can continue the conversion process in a new cell. The prion can therefore propagate as well as replicate. If eaten by another susceptible host, these newly misfolded prions can transmit the infection from organism to organism. Coming to the function of prions, the normal prion protein PRPC helps in activation of myelin repair in Schwann cells of peripheral nerve fibers. So, lack of PRP proteins causes demyelination in those cells. It is also suggested that PRP may have a normal function in maintenance of long-term memory. PRP expression on stem cells is necessary for an organism's self-renewal of bone marrow. Hematopoietic stem cells express PRP on their cell membrane, and those hematopoietic tissues with PRP null stem cells exhibit increased sensitivity to cell depletion. It is also shown to have molecular signaling function. Next one is amyloids. Amyloids are aggregates of proteins that become folded into a shape that allows many copies of that protein to stick together, forming fibrils. Amyloids is form of long unbranched fibers that are characterized by a cross beta sheet quaternary structure in which parallel chains of beta stranded peptides are arranged in an orientation perpendicular to the axis of the fiber. Amyloids are formed by aggregation of the misfolded normally soluble proteins. Coming to amyloid pathology, amyloidosis is the group of diseases in which the abnormal protein known as the amyloid fibrils builds up in tissue. Amyloid fibers deposit and accumulate to form plaques around cells and disrupt the healthy functioning of tissues and organs. They play a role in neurodegenerative disorders. Some amyloid proteins are infectious. They are called prions. The reason for amyloid-associated diseases are unclear. Calcium dysregulation has been observed in cells exposed to amyloid oligomers. Amyloid deposition is associated with the mitochondrial dysfunction and a resulting generation of reactive oxygen species ROS, which can initiate a signaling pathway leading to apoptosis. Normally, misfolded protein are degraded intracellularly by proteosomes and extracellularly by macrophages. But in amyloidosis, the quality control mechanism fails. So, there is a rise in the amount of amyloid proteins. This table shows the disease featuring amyloids. Some common ones include Alzheimer's disease, diabetes mellitus type 2, bovine spongiform encephalopathies, atherosclerosis, rheumatoid arthritis. There are many organs affected by amyloid deposition like brain, heart, spleen, liver, pancreas and kidney. Now, it would be interesting to know that certain amyloids are not involved in any disease state and they have certain functions in the body of the organism. For example, protein or peptide hormones are stored as amyloids within endocrine secretory granules. There are functional amyloids in Pseudomonas, and amyloids are highly abundant in many microbial biofilms and play an important role in their architecture. I would like to conclude by saying that the proteins, enzymes, prions and amyloids are examples of special proteins as these have certain special characteristic features. Enzymes are monoclonal antibodies that function as enzymes. It may be possible to generate enzymes with the ability to dissolve blood clots or to cleave viral glycoproteins at specific sites, thus blocking the viral infectivity. They also contribute to some autoimmune diseases. Prions can be considered as infectious amyloids. Amyloids being an accumulation of compact and cross-linked beta sheets, they form plaques around tissues leading to neurodegenerative diseases. Few amyloids are involved in disease state while few others are necessary for normal body function of the organisms. Thank you.